The new CRISPR Lab, developed in partnership with Bio-Rad, introduces students to cutting-edge molecular biology. Students use the Bio-Rad out of the Blue CRISPR Kit to edit bacterial DNA and turn bacterial colonies from blue to white. They do this by disrupting the function of the LAC-Z gene, which normally breaks down the substrate X-gal to produce a blue pigment. First, students are provided with background information on general gene editing principles and the CRISPR-Cas9 system. They learn about the LAC-Z gene, the role of arabinose, and plasmids in the editing process. The challenge page tests students' basic knowledge before they begin the lab protocol. Students are asked to label the Cas9 sgRNA complex and are tested on their understanding of key concepts, like LAC-Z gene function and the role of arabinose. Importantly, students are asked to make a prediction as to how their actions will change LAC-Z gene function and the color of the resulting bacterial colonies. Students use the CRISPR kit and a clearly illustrated procedure to edit bacterial DNA. During the analysis, they count the number of different colored colonies in each plate and explain their observations. Students are asked to make a claim, provide evidence and their reasoning, and rebut alternative explanations. On the review and integration page, students' learning is consolidated as they review their predictions, discuss why controls are necessary, examine a use case of CRISPR called camera, and reflect on what they'd like to investigate next. In the first optional extension activity, students choose a genetic disease and perform a blast search to find a relevant target site for CRISPR editing. In the second extension, students confront the ethical considerations of CRISPR. Students are guided to summarize their work in a scaffolded lab report before completing the lab. Thanks for looking through one of our new biology labs, created in partnership with Bio-Rad. We hope you've enjoyed learning how you can bring cutting-edge molecular biology techniques to your students.